Good evening, Olivia and Ashwini. We'll start the session now and then let others join. I hope I am audible and the screen is visible properly. Can you just let me know in the chat box once? Is the audio clear? Oh, okay. So there were a few questions that was left in the last session from the last week's content. A few basic questions. We'll first go through those questions. So can you tell me what is the need for environmental biotechnology? And before that, if you have any specific doubts from previous lectures, you can also post that in chat box. If not, please try answering this question. Okay, initially I have few questions left from what I prepared for the previous class. So I'll just discuss those questions. So the first thing is about what is the need for environmental biotechnology. So the main needs include, yeah, okay, there is a response. It applies biological processes to solve environmental issues and challenges like pollution, waste management, obtain sustainable energy, okay, mitigate climate change, and public health. Right, almost everything is covered in this. So, first of all, we need some sustainable solutions and we need to improve environmental quality. Like you told, we need to solve environmental issues and challenges like pollution and waste management and security. So security in terms of uh, it can also be related to the sustainable energy production that you have mentioned, and finally human health. So how are we going to achieve these using environmental biotechnology is that we are mainly going to use microorganisms, and these microbial communities uh, know multiple mechanisms through which they can survive in extreme environments, and they are able to organize and sustain themselves in all the extreme environments. So if we are able to decode the mechanisms by which they are doing it, then we can um, apply the same for the current environmental problems as well. So the next question is, what are the resources which is of the focus of environmental biotechnology? Primarily, this does not have any single correct answer. So there are multiple options you can try giving them. Okay, solid waste is one resource. Any other resource? Okay, right. So mainly water, energy, and wastes are the resources that environmental biotechnology is mainly focusing on. So the next question is, the principle of DASH emphasizes tracking redox flow through microbial pathways to understand the community metabolism. This is just a memory based question. Okay, there is one answer for redox equivalence. Anyone else want to try? Okay, so the answer is redox equivalence or the principle of follow the electrons. It's very commonly known as follow the electron principles. To backtrack, redox equivalence, redox balance, everything is right here. So the next question is kind of a short answer type question. Like based on your understanding, can you explain how multi-scale structure in microbial ecosystems influences the design of biotechnological processes? Like we are going to study the existing microbial ecology in a particular environmental condition, and then we'll try to extrapolate such system for remediation or treatment processes. So how are we extrapolating this multi-scale structure in the design of biotechnological processes? Just a one or two sentence answer would do. If anyone wants to unmute and answer, that is also fine if the question is big. I mean, if your answer seems to be comprehensive, you can also unmute and answer. Okay, there is one answer that a multi-scale structure involves genes to cells to population to communities. Okay, that's a nice way of explaining multi-scale structure. So when we are studying it, it allows bioengineers to optimize microbial mixtures and scale up processes and control conditions for high output. Right, okay. Any more answers? Okay. So the answer that I had was the same. Multi-scale structures range from genes to cells to aggregates to communities. And these structures usually dictate how the nutrients are present in the environment and how they are consumed, because the rate of consumption and the rate of the other product protection influences the gradient of these nutrients, and also species distribution. Like if the environment has some difference in the nutrient availability, based on that the species distribution will also vary, and also depending upon the interspecies interactions the species distributions can vary. So as we have learnt about this entropy and all, if one organism depends on the product of the other organism, then they are hierarchically present even in the environment. So all this knowledge we can get, and after that there should be a process design that accounts for all this. So we have to have a process design in which if at the first stage uh, some organic matter should be consumed, and then in the second stage the product of it is consumed, we have to design a process that will allow the first step to happen first, and then followed by the second, third, and so on. So this is how this multi-scale structure in microbial ecosystems can influence the design of processes. Another question, like this is simple question, you can just give what is synergistic and antagonistic, and if you have or if you know any example you can also state that. If not, it's also fine without an example. uh, I just want you to know what is the difference between synergistic and antagonistic interactions. Okay, an example for synergistic is, so the meaning of synergistic is helping each other. Example is fermenting bacteria making hydrogen, which methanogens can use to make methane. And for antagonistic, they are okay, opposing or hurting each other. Streptomyces kills other microbes by making antibiotics. Right, perfect. So synergistic interactions enhances the so as they are helping each other, on the whole it enhances the community productivity. Whereas this antagonistic interactions, because they are kind of competition where one organism tries to overpower other organism, it can create discrete clusters, and it helps to maintain diversity. Good. Okay, this is the next question. Name the energy carrier outlets from organic matter. Um, you try naming few and then we'll see the answer. So after week four's lecture, there can be like multiple kinds of answers for this question, like that I can expect from you, but still, I'm not asking energy carriers, energy carrier outlets from organic matter. This is in the context of organic matter to energy generation. So what can be the energy carrier outlets that can be generated from organic matter, to make the question simple? Okay, as I told you, I've given both the kind of answers that I had expected. So, uh, the first set of answers that you have given, the ATP, NADH, and FADH, are general energy carriers or within the cell. So, as I explained, the question is about energy carrier outlets from organic matter. So, in that case, your second answer is perfectly the right answer. So biomethane, hydrogen or biogas, bioelectricity, ethanol, and you have also added heat. So all these are the energy carrier outlets from organic matter. Nice. So the next question is kind of match the following question. And others in the meeting, I also encourage you strongly to post your answers. This is just a meeting with very few people, so you need not like hesitate to answer. So you can just post your answers in the chat box. Okay, so based on one answer, we match fish with specific gene, PCR with total RNA and DNA, and metaomics with rRNA. And based on the second answer, we are going to match fish with rRNA, PCR with specific gene, and metaomics with total RNA and DNA. Anyone else in the meeting wants to answer this question or wants to go with any of the previous answers that is given? Okay, so the answer is actually fluorescence sensitive hybridization can work with rRNA. Like we can hybridize or we can make fluorescent tag probes for rRNA and then visualize it. That is possible. But PCR is generally gene specific. Like because in PCR the most important part is the attachment of primer. So when we are going to design some primer, then it means that we are going to target some kind of gene. Even in all the PCRs that we are talking about and all this omics technology and all, we will be having some conserved region. So we are targeting those conserved regions to develop the primer so that we try to target most of the uh, organisms that can be present in the mixture. It is going to be conserved among the species, so we are trying to target specific genes in PCR. And in the case of metaomics, we know that if we are going to deal with the term meta, it is going to be complete or total. So that is why total RNA and DNA is matched with metaomics here. I hope it is clear. Like if you want more details about this PCR technique or something, you can let me know the chat box so that I can discuss that. So this question is kind of subjective. What can be the goals for the new molecular tools for microbial community analysis? High through it should like these are not the options. I'm just directly displaying the answers. It should be high throughput. They should be quantitative answers that we can derive from those tools. And the third thing is it should elucidate the structure and function simultaneously. We currently have tools to analyze microbial community, but if we can improve the tools further or find new molecular tools, we should ensure that we mainly target high throughput analysis and obtaining quantitative results and also obtaining structural and functional information simultaneously so that we save time and resources and we can get an holistic answer. Okay, so the next question is name the techniques used to study nutrient cycling. Need not be only from the lectures. I have given the answers from the lecture slides only, but if you have any other answers, you can also post this. Okay, one Olivia has answered that we can use metagenomics, PCR, fish, GC. Do you mean chromatographic techniques or okay, gas chromatography, bioreactors, and enrichment cultures or sensors? Okay, partially it is possible. Like in case when we use metagenomics, maybe metagenomics cannot exactly tell us. Maybe we can try to speculate, like if some kind of sulfur reducing bacteria or nitrogen fixing bacteria is present, some nutrient cycling related to that can be present in that environment, that we'll be able to speculate, but I mean we will not be exactly able to study. Maybe when we use something like a proteomics or metabolomics, something on that line, then maybe we will try to monitor what kind of genes are expressed, or metaproteomics, metatranscriptomics can tell us what kind of gene is expressed or what kind of enzymes are produced. So based on that also we can try to speculate. uh, and then GC, PCR and fish, it is mostly to trying to deal with the DNA of microorganism, and it is not a technique that directly gives us the details about nutrient cycling. It can help us in the study of that environment or the microbes, or the study about the microbes in the environment, but it cannot directly tell us about the nutrient cycling. um, so in the case of gas chromatography, uh, I think you're talking about all those pathway that we will be able to predict with gas chromatography. So when we are able to identify all the metabolites that are available, in that way GC can be used. Bioreactors also the same way. Enrichment cultures, like we are going to give specialized conditions and monitor what is happening. But even then, we'll be able to, in case of enrichment cultures, if you are going to do a pure culture, you will be able to identify the potential of a particular microorganism, but as a whole how nutrient cycling happens in that ecosystem will not, we will not be able to decipher it properly. Okay, so I think this possible genes that suggest a particular pathways in regard to this metagenomic approach that we were talking about. So everything that you have mentioned here is a little indirect way through which we can decipher the nutrient cycling. Can anyone think of any direct methods that? So as I told, I have answers only from the lecture slides. So are any of you able to think of any answers? I'll just wait for one more minute and then I'll display the answer. Okay, so exactly, it is stable isotope probing and nanosecondary ion mass spectrometry, which is nanosims, or SIP and nanosims in their short forms. So when you talk about this, both of this, we are going to label one of the nutrient. Like if I'm going to study carbon, right, isotopic uh, tracking, or not microelectrodes, but isotopic tracking we can do. So the nutrient that I am concerned of, let's say carbon, so I will do some kind of isotopic probing and check and tag that carbon, or I'll use a isotopic carbon which is different from the normal carbon that is present in that ecosystem. So when I use methods to detect that special isotopic carbon, then I can see how the carbon flows in the system. Like I initially give some tagged carbon in the substrate and see how that carbon is being used by the microorganism, and then where and all it is getting passed. So based on this, I will be able to draw the nutrient cycle chart using stable isotope probing. Similarly, in the case of uh, nano nanosecondary ion mass spectrometry, I'm not going to tag anything, but maybe if I'm going to bombard this in my sample with some beam, I can get to know the elemental composition. So when I'm going to do it repeatedly, I'll be able to track how the nutrient is being cycled in that environment. So these two can be a little bit more direct and specific than the techniques that you have mentioned in the chat box. So initially we can try to study the microorganisms and speculate some pathway, and using the techniques that I have mentioned here, we can try to have a confirmatory study if things happen the same way as we have speculated. So the next question is to identify the true statements. Okay, as rightly answered, both A and D are right. Biomass is a storehouse for electrons, and methanogenesis is one way of recovering energy value of BOD. Whereas dissolved oxygen depletion occurs during low BOD consumption. No, when BOD is rapidly consumed, then oxygen is utilized, and therefore dissolved oxygen depletion occurs. And oxidized contaminants are not similar to BOD. They are in fact kind of opposites to BOD, because whatever thing that has the potential to be oxidized, for that we tell that there is some biochemical oxygen demand for that. So in that case, oxidized contaminants are not similar to BOD. And methanogenesis is one way of recovering energy value of BOD. As we saw a few questions back about that energy carrier outlets from organic biomass, like biogas or biomethane is one way of recovering the energy value. And The consumption of COD or BOD happens in the reactor, and at the end we get the biogas. So, right, all your answers are right. So, the next question is identify the organism with high growth rate among the three organisms. Aerobic heterotrophs are nitrifiers or methanogens. Which can grow faster? Exactly. So, aerobic heterotrophs have higher growth rate compared to nitrifiers and methanogens. Nitrifiers and methanogens are very slow growing organisms. So, from this question onwards, it is from the week 4 lectures. So, this is again an assertion and reasoning question where the assertion reads metagenomic analysis should be PCR less to avoid bias in community analysis. The reason is PCR primers may not work uniformly for all genes they are designed for, leading to selective amplification. Okay, three of you have answered. So your answers are right. So metagenomic analysis should be PCR less to avoid bias. As we saw in one of the questions, PCR is very much gene specific. So if the particular organism in our analysis, metagenomic analysis, doesn't contain the gene for which we have designed the primer for, then it's going to be problematic. It is not going to be amplified. So in that case, it leads to a selective amplification, uh, and hence PCR less techniques can avoid bias in the community analysis. This is the next question is metatranscriptomics provides real-time data about gene expression in microbial communities. And the reason is mRNA has a longer half-life compared to DNA, making it stable for analysis. So Ashwini, do you have any answers to give or shall I proceed? Okay, as you have mentioned, the right answer is C. Metatranscriptomics indeed provides real-time data because mRNA is generated, and it whenever a gene has to be expressed, mRNA is generated, and then the half-life of mRNA is very short. So once there is translation to protein, mRNA is then being depleted. So uh, metatranscriptomics indeed provides a real-time data, but the half-life is very short, so it is not stable for analysis. We can get instantaneous results, like at a particular time point what is the gene expression that is present, we can study using metatranscriptomics, but it cannot give us a very stable or long term data. So the third assertion is that environmental biotechnology research should be theory driven rather than tool driven, because tool driven research leads to stamp collection of data without meaningful biological results. Okay, one answer is D, one answer is A. Can you differentiate between theory driven and tool driven research? Just a one line definition of both. Okay, so the right answer is A actually. Uh, why? Because the difference between theory driven and tool driven we'll first discuss. So theory driven is uh, See, whenever we are planning some research work, we'll be planning a set of experiments to be done. In case of theory driven research, we will have a hypothesis in mind or a final destination in mind. Like, I have to check whether this is happening or not. Or that will be the question. So that will be the theory that will be driving our research. So you will be planning for experiments which will lead you towards that answer. So that is how theory or process driven research will work. In case of tool driven research, you're going to do something because you know there is the availability of that tool. So you know that metagenomics is present, and that is why you do some genomics, and you know that proteomics is present, and that is why you're going to do some proteomics. That is tool driven research. When you have the knowledge and ability to do something, you do it. In case of theory driven, you are given a place to identify, okay, what can be the probable um, kind of metabolism that you expect to happen. So in that case, what you do, you should do some genomics and identify some bacteria that are present there, and you also do some proteomics and identify some genes that are expressed in that genes that are expressed and the proteins that are present in that environment. So then you correlate both, and then you speculate that, okay, these organisms and are present and they have expressed these proteins also. So this could be the probable mechanism that is happening in that environment. So you have some research question in that way, and you answer it in a um, experimental way, which means you are satisfying a theory. That is a theory driven research. So in this, environmental biotechnology research should be theory driven rather than tool driven is right. Why it should not be tool driven? Because tool driven research leads to stamp collection of data without meaningful biological insights. So stamp collection means at a given point of time you have some sample in hand and you have a technique which you can use, and just because you have both, you are doing some experiment randomly, but you're not trying to have any network or any connective analysis and get some meaningful insights. So stamp collection, when we call or tell about stamp collection, it is just at a particular given day and time you have some sample and you have done the analysis for it. But when will you get meaningful biological insights? When you do it for a little longer period of time, just to have some confirmation or to account for seasonal variations or something like that. You will do it for a little bit longer period of time, like a monitoring study. Right. So in that case, tool driven research leads to stamp collection of data without meaningful biological insights, and so this is a statement. And because of that, environmental biotechnology research should be more oriented towards theory driven rather than tool driven research. I hope it is clear now. So from these, identify the false statement. I am missed giving the options here. Just take the first one as A, B, C, and D respectively in this. So the A statement would be macroecological theories can be directly applied to microbial ecological systems. B statement is metabolomic studies reveal the ultimate functional response of microbial communities. C statement is spatial and temporal scales pose significant challenges in environmental microbiology research. And the D statement, or the fourth statement, is metaproteomics validates the findings from metatranscriptomic studies. Okay, both of you have answered. I'll just wait for one more minute to see if anyone else answers and then we'll discuss. Okay. So, as you have answered, A is a false statement because macroecological theories means theories that are pertaining to macroorganisms in the ecosystem. So, macroecology deals with macroorganisms and their interactions rather than microorganisms. So what happens is when we can't apply these macroecological theories because microbial interaction is very spontaneous, and also all reactions happens at a very micro scale. The interactions are very rapid, spontaneous, and hence all these theories cannot be directly applied to microbial ecological studies. And for the other cases, so metabolomic studies reveal the ultimate functional response of microbial communities. So as a whole, a community gives a functional response to the kind of environment it lives in, and metabolites or metabolism is the utilization of some resource and the building up of some cellular structure. So it has two components. Metabolism has anabolism and catabolism. One is breaking down of resources, and another one is building up of the cell. So on the whole, all the process that happens together, we term it as metabolism, and the study of the metabolism is metabolomics. So this gives a fair enough idea of the complete functional response of the microbial community. And in the next point, spatial and temporal means space and time based changes are very significant in case of microbiological research because these microbes are constantly evolving and they are very spontaneous. So at a given space and at a given time, our findings can be right. So we have to carry out the research at multiple spaces and multiple times to come to a larger conclusion and to understand the dynamics of the microbial community. And also metaproteomics validates the findings from metatranscriptomics. Why? Because through metatranscriptomics, we will try to decipher what genes are expressed. So if these genes are expressed, or if this mRNA is produced, then this kind of proteins can be present, and this kind of functions through the proteins are expected to happen. That is what metatranscriptomics tries to tell. So while doing metaproteomics, we can try to validate, okay, I speculated that this kind of transcription or transcripts can lead to such a function. So that can be again validated by metaproteomics when we confirm that that protein was actually present and working in the same form. So the next question is simple question, very simple question. What is the total DNA extracted from environmental samples without isolating specific organisms called? I hope things are clear. If it is not, please let me know in the chat box. Right, it is either a metagenomic DNA or an environmental DNA, as people generally call it. Okay, so another question. Molecular interrogation or investigation of microbial community should be dash dash and dash to gain maximum insights. So this is a very open-ended question. So whatever comes to your mind, you can just answer. This was also discussed in one of the previous slides today. Or based on your understanding, how should molecular interrogation of microbial communities be? Okay, interesting answer. It should be genomic transcription and proteins. Like we should study about DNA, RNA, and proteins together to get maximum insights. Okay, that is one interesting answer. Any more answers? Okay, so because there was only three blanks, you gave three, and metabolism can also be included. I had a very different kind of answer, so that is why it was three blanks, but all your answers were also equally right. So, the answers that I was able to prepare included uh, the approach should be multi-directional. So, like you told, genomics, transcriptomics, proteomics, and metabolomics all together. So, it should be a multi-directional approach. It should be high throughput so that it is highly efficient for us to get information and analyze, and then it should be quantitative. The next question is the integration of omics data requires essential support from dash tools for database management and data mining. Uh, so here the answer is another stream in biotechnology that many of you would have been introduced to. What is the field of study that we can use to integrate all the omics data? Exactly. So bioinformatics is the field that we require. And another question based on previous discussion, you can answer it soon. What is the term stamp collection in molecular microbial ecology refers to? Exactly. So stamp collection refers to gathering data without a theoretical framework. So that is why we are not like very much interested in tool-driven research. So what is the term perennial moving target refer to? Chan- uh, does it mean changing sampling locations or rapid microbial evolution or seasonal variations in microbial activity or migration of microorganisms? So right, perennial moving target refers to rapid microbial evolution. So when we are trying to evaluate the role of bioinformatics in omics. So, as one of the questions we discussed was based on this. So, for those of you who are new to this bioinformatics, so first of all, it gives us a database support because we'll be getting large sets of data when we are going to do some genomics or something. It's going to be for the entire microbial population that is present in our sample. So, we'll get huge amount of data. We have to manage and mine this data. So in that case, database support will be given. And next is about the data integration algorithms. So as we talked in the earlier one of the questions, we need to have a holistic approach. We need to have data from genomics, transcriptomics, proteomics, and metabolomics. And finally, we have to have a network connection between all these different techniques to have a holistic understanding of the ecosystem. So data integration algorithms is also provided by the bioinformaticians. And next, they also give statistical tools for identifying the patterns across different omics layers. So this is part of the data integration. And finally, we have network analysis tools to understand the pathways and species interactions. And finally, there is this visualization tools that makes it easier for us to represent the data and makes it easier for others to understand what exactly is happening in that particular ecosystem. And finally, based on whatever data we already have, we can use some machine learning approaches to build a predictive modeling. So I have gathered some enough amount of data from a particular ecosystem that if A is the condition, uh, the ecosystem is going to behave a certain way. If B is the condition, it is going to behave a certain way. And hypothetically, if I am going to build a model and ask if some X or Y is the uh, nature of the environment, then how will be the behavior of the ecosystem. So in that case, we can use machine learning based predictive approaches. And finally, all this gives us long term stewardship and maintenance of the data, so that we get the maximum information out of the data. So generating data is one thing, and deciphering the inferences out of the data is also a very important thing in environmental biotechnology. Only based on the information that we are able to decipher from the data, we will be able to develop processes that are going to benefit the environment. So in that case, this transition is likely very important. So the next question is about microorganisms. So phototropic microorganisms can utilize light as an energy source in environmental engineering systems, and phototropic organisms rely exclusive on inorganic carbon sources for their metabolism. Like you just have to tell if both these statements are right or if any of it is wrong or what. So we have B as the answer, but it is C because so uh, according to you, both A assertion and the reason are right, but it is not the right explanation of A. But no, R is not right because phototrophic microorganisms do not rely exclusively on inorganic carbon sources for their metabolism. There are certain phototrophic microorganisms that can use light as energy. They have these pigments, so they can harvest energy from the light source, but they do not rely exclusively on inorganic carbon sources. They may depend on some acids or alcohols that are present in the environment. So there are organisms that can do this, like purple sulfur bacteria or uh, few bacteria like that. They can harvest light energy, but they do not rely exclusively on inorganic carbon source for their metabolism. So usually we directly tend to have a notion that if the organism is able to harvest energy, then it is only going to consume carbon dioxide for to produce its own food and to satisfy its metabolic needs. That is not the case. It can also depend on some organic carbon sources, and that is why the answer is C, not B here. uh, Phototrophic microorganisms rely on inorganic carbon sources for their metabolism is a different statement, and rely exclusively on inorganic carbon sources is a statement that gives a very different meaning. So that is why the answer here is not B as you told, but it is C. So from these four statements, identify the false statement. So the first statement is facultative anaerobic bacteria can grow in both the presence and absence of oxygen. The dominant microbial process in acid mine drainage is lithoautotrophy. The metabolic versatility of facultative anaerobes provide them with ecological advantages in fluctuating environments. And the autotrophic microorganisms fix carbon dioxide as a carbon source and require reduced organic compounds as electron donors. Which of these statements is or are false? Okay, so D is the right answer. So facultative organisms are those that can live in kind of both environments. Like if we say facultative anaerobes, they predominantly can live in anaerobic condition, but they will be able to tolerate some amount of aerobic conditions. When we talk about facultative aerobes, then it's the opposite of that. They predominantly prefer aerobic conditions, but can also survive in a little bit anaerobic condition. Not too much of that, but they'll still be able to survive in that conditions. That is facultative organisms. So in that case, it can grow both in the presence and absence of oxygen is right. And the third point is in relation to the first point, where the metabolic versatility of these anaerobes makes them uh, undergo this shift in case of environment fluctuations. Whether there is presence or absence of oxygen, they have metabolic versatility to adapt to both these situations. And in the second point, the dominant microbial process in acid mine drainage is lithoautotrophy, because in acid mine drainage, uh, if there it is going to be autotrophic, the electron donor is going to be inorganic. So lithoautotrophic process generally takes place in acid mine drainage. But in the case of last point, autotrophic microorganisms are going to fix carbon dioxide. Okay, we know that they can utilize carbon dioxide and they are going to fix it in the form of glucose or products like that. But um, they mostly do not require only reduced organic compounds, but they can also survive on sulfates, nitrates, etc. So reduced inorganic compounds can also act as electron donors in the autotrophic microorganisms. In that case, that is the false statement. So the next question is again an assertion and reasoning question. Oxygen is the most favorable terminal electron acceptor in microbial metabolism because the redox potential difference with oxygen is the highest, and hence we are getting maximum energy yield. Are both the statements true, or is either of it false? Okay, so the answer for this question is A. Oxygen is the most favorable terminal electron acceptor in microbial metabolism because the potential difference with oxygen is highest. So there is a concept of microbial redox tower in microbiology, or in case of environmental microbiology, it is like very important. So when you imagine a tower, uh, this is going to be a redox tower. So whatever are the species that are present on the top of the tower, they are going to have a higher electronegative values, and the reduced form of these are going to be electron donors. When you move towards the bottom part of this tower, that will have a more positive electronegativity value, and the oxidized form of these are going to act as strong electron acceptors. So uh, bo- the species that are present on the top as well as the bottom, they are a redox pair. So as we all know, redox reaction is a continuous reaction. Whatever gets reduced can be oxidized and vice versa, right? So it is going to be a redox couple. So the redox couples that are present on the top of the tower have higher electronegativity value, and in the reduced form they can act as electron donors, whereas the bottom of the tower, since they have the positive e naught value, they can, in the oxidized form, they can act as good electron acceptors. So ideally the direction of electron flow is going to be from top to bottom. So in this case, from when oxygen is going to be the uh, electron acceptor, it is going to be at most bottom of this electron tower compared to other electron acceptors that can be present in the environment that can help the microbial metabolism. So that is why, since the distance in the tower is higher, we call that as a higher redox potential difference. So when more potential difference is covered for the electron to reach the oxygen, in the intermediate steps, there is maximum ATP yield, so which we correlate to maximum energy yield. So this is the explanation for why oxygen is more favorable terminal electron acceptor in microbial metabolism. So uh, if you are interested in this, to understand this part, you can read about electron towers, or you can also drop a text here so that I'll show you the electron tower and explain what it is in the next class. We'll go for some 2 to 3 more questions before we end for today. So the next question is, identify the false statement. The first statement is that the mixed microbial consortium cannot stably coexist in engineered ecosystems. Obligately anaerobic bacteria, it is not ac, I'm really sorry for the typing mistake. Obligately anaerobic bacteria lack protective mechanisms against oxygen induced radicals. The third point is, increasing redox potential increases the amount of ATP produced during respiration. And the fourth point is, redox potential for electron acceptors determines the energy yield of microbial metabolic processes. Which of these is false? Okay, so we have two answers, A and D. So A is the right answer. So mixed microbial consortia, they can stably coexist in engineered ecosystems as well, because we have mixed organisms in that. So whatever is able to survive in the engineered ecosystem that we are providing, that will get an upper hand. It doesn't mean that it is not going to stably coexist. The stability is finally going to be maintained. And obligate anaerobic bacteria, in the sense it can live only in anaerobic condition. Even little amount of oxygen or air can disrupt that microorganism. So in that case, why? Because they lack the mechanisms against oxygen induced radicals. And when the redox potential difference is increasing, the amount of ATP produced is also increasing. Like I told, the concept of electron tower is very critical in understanding this. The same with the fourth point also. In the case of electron acceptors, so whatever is the potential difference, that is going to determine the energy yield of metabolic processes. So maybe before starting the next lecture, I will once show you the redox tower and explain how it works. So imagine a tower, and in the bottom of the tower, you are going to have all the acceptors. Okay, so the last acceptor will be your oxygen. So before that, you can have two to three electron acceptors. If let's say in the acceptors you have uh, you name the bottommost one as 3, and before that there is 2, and before that there is 1. If 1 is going to be the electron acceptor, x amount of energy is going to be produced, and if 2 is going to be the electron acceptor, y amount of energy is going to be produced. The y amount of energy that is produced will be greater than the x amount of energy, because it is traveling furthermore in the tower, and hence the energy yield will be more in the process. So that is why that statement is true and not false. So another question, I guess in the first two lectures somewhere I mentioned what are methanogens. Methanogens are a group of dash that use carbon dioxide as electron acceptor under strict anaerobic conditions. So they are going to reduce carbon dioxide, that we all know, and they live in strict anaerobic conditions, that is also we all know. What are these? What kind of microorganisms are methanogens? Exactly. So they are archaea. So initially we had discussed about bacteria, archaea, fungi. So generally we many would be clear that fungi, algae, and bacteria are different, but mostly we tend to confuse bacteria and archaea. Bacteria is different and archaea is different. Archaea has a cellular architecture that enables them to live in extreme conditions. So initially they were identified only at extreme conditions, and all the cell wall composition was and all, different for archaea from bacteria. Another simple question based on this redox reactions only. Sulfate, nitrates, and ions can be examples of dash in microbial respiration. Right. They are electron acceptors. So this question, I'm just displaying it now, but maybe you can answer it in the next class also. I'll just present it as the first question in the next live session. Which process yields the least energy in microbial metabolism? And you can note down the question as well as the options. Maybe you can check out which can be the answer for this with the proper uh, explanation of why. So the next question, that was just postponed for the next class. So the next question ideally is, in the absence of suitable external electron acceptor, microorganisms use which process out of the following? Okay. Any guesses on why fermentation is preferred? So it is right that fermentation is mostly preferred. Okay, so let me give the explanation. Okay, gives least ATP compared to respiration under strict anaerobic condition. Okay, and the next answer is fermentation allows microorganisms to regenerate NAD plus without the need for external electron acceptor. That is the major part. So we all know NAD and NAD plus and NADH is a electron carrier pair. So in the reduced form it is NADH, and it is when it is oxidized it is NAD plus. So ideally in the case of microbial metabolism, this NAD plus is reduced to NADH, and this has to be cyclic. Again, this NADH must be oxidized to NAD plus. For that is this has to be regenerated for the process to continue. When we don't have proper external electron acceptors, the regeneration of NADH is not going to happen, which means the entire upstream is going to be blocked. So there is not going to be further conversion of NAD plus to NADH and all, because already whatever is present in the reduced form. Only when it is regenerated we can further go for continuing the process. So in that case, the fermentation ideally helps in regeneration of NAD plus from NADH. So the proton associated with NADH is given to the fermentation process or the fermentation products, and hence NAD plus is again regenerated, and finally the upstream processes also continue happening. So that is the exact reason. And one more last question for today. So a very simple question, just match the following. So with this question we will close today's session. Okay, so ascomycetes, all of them are microorganisms. Ascomycetes have spores contained in a sac called ascus. Euglenophytes are photosynthetic algae with a flagella, and basidiomycetes produce large fruiting bodies, like, for example, mushroom. So I also have a couple of other questions that follows from this, from exact microorganisms. So maybe we'll discuss all these questions in the starting of next class, and then go with questions with week 5 lectures. Um, so with this, I'm closing today's session. If you have any doubts, you can post in the chat box, or you can leave today's session. And thank you for participating. Though we had only less number of participants today, whoever was present was really active. And as always, like, my sessions only depend on question and answer, so I need responses from the audience side. And thank you for being so attentive. Hoping to see you all next week also. If you have any doubts, please post in the chat box or unmute yourself and ask. Thank you, Ashwini. Sheetal, do you have any doubts or can we close the session? Okay, thank you. Thank you, Sheetal.